Okay, hello, ladies and gentlemen. This is Double Education, a channel of education who's presenting different channel of educations, biology for grade 9, 10, 11, and 12. Now, this Double Education is going to present pre-university biology remedial for remedial students. So please don't forget to like, subscribe, and share your channels, Double Education Centers. The presenter of this channel is Mahfouz Muhammad, your teachers from Jiga University Biology Department staffs. I hope, as you don't forget, like, subscribe, and share your channels. If you have comments and suggestions, you are put comments on my channel or my video, then I will correct and also present what you need new topics. Okay, as you remember, this remedial program is presented for the students who score under half of the national exams, but all of the follower or attending of the remedial program is the winner one because you got these results by your own efforts. I hope as you win and you pass to now freshman programs by 100%, you have a chance. If you do something good and try your best, you make chance to change. Make chance to change chance. This C and the G letter is always my slogans. Okay, don't forget try to share Double Education Centers, subscribe and follows. And also give me the topic you need and not clear for you, then I will present in this Double Educations. So the course title is Biology Remedials, as if it's presented by Jiga University Biology Department staffs. Unit 1 says about science of biology. As you remember, this remedial biology content have 10 units. The first unit is science of biology. Those you are studying previous grade 11 unit 1. In this units, we are going to talk about what is science, what is biology, different area of biologists, different scientific methods, steps, different scientific reports, steps, different variables, cause and effects of researches, dependent variables, independent variables, controlled variables, confounding variables, different accuracy, reliability, and everything. Those materials or tools of biology that biologists use in laboratories, and the tools of biology that biologists use in fields. Let me see, starting one by one. Unit 1 says science of biology. This science of biology, let me start by defining biology. So biology is the science of life in the living organisms. It's the science of life in the living organisms. It's come from two Greek words. Bios means life, and the logos means study. So generally, the biology is a branch of natural science, those study about the study of lives. Generally, biology is a study of lives. So biology is the science of life in the living organisms. Generally, if they talk about study about life in the living organisms, it is study about unicellular and multicellular. Unicellular means the organisms was made from only single cell or one cell. And the multicellular means the organisms made from more than 2 cells or many cells. Good. Let me see different area of biological studies. We says biology is study about living things. So there is area of biology of studies. The first area of biological studies, astrobiologists. Astrobiologist means a person or a branch of science that try to find evidence of life on other planets in our solar system. They study evidence. Is it a living organism living in another planet or in galaxy or elsewhere in the universe? They try to find evidence. Acid another living organisms living in another planet other than Earth's. This is what astrobiologist studies. The second one is biomedical research. This biomedical research is a branch of biology. It depends study about health, development of new drugs, development of vaccines. It study and talk about development of new drugs and the development of vaccines. It also study the way in which this is developed to gain better understanding of them so that the cure can be found. The better understanding of disease, then study about how we course from this disease by finding drugs, by finding vaccines, by finding different diagnostic methods. This is all about biomedical researchers. I hope it's clear for you. When we come to the third one is microbiologist. It's come from two words, micro, which means small, biology means life. So a study about small life, or very small life, which means microorganisms like bacteria, virus, fungus, etc. So it is totally a study about how microorganisms of all kinds functions, its characters, styles, behaviors, its beneficiary factors, sides, and also pathogenic sides. They talk about these microorganisms. The another branches of biology is also paleobiology. This paleobiology is the way in which life began on Earth, how life is begun on Earth, and how it has evolved from simple life forms into more complex one by studying fossils. Branch of biology study about fossils and they study about evolution of life previous. And they use evidence from rock fossils to estimate when and how new life is formed. It's appeared on this planet. By studying life, let's talk about when and how new life is formed. This is about the study of paleobiology. Not only this, besides these other biological studies, biology may include a lot of branches. Besides this other biological study, biology may include different uh, areas. It's maybe medical doctors, dentists study about teeth, veterinary surgeons study about animals, physiologists study about physiology of animals and the human things, geneticists, oncologists, parasitologists, neurobiologists, branch of biology study about brains, entomologists, branch of biology study about insects, physiotherapists about physical therapy of human beings, ethologists about tissues, zoologists about animals, botanists, and also nurses, another area of biology. Not only this, we have many other areas of biology. Let me come to defining science. What is science? As you remember in grade 11, the word science is derived from Latin words, which is called scientia, which is to mean knowledge. The word science is come from Latin words. It's called scientia, which is mean knowledge. It's a unique system of acquiring knowledge. Science means in another ways unique system of acquiring knowledge based on scientific methods. It's a way of best way or another unique way of acquiring knowledge, but we are only depending on scientific methods. It is sometimes called biological science is called experimental science. It's depending on experiments to develop theories, to find evidence, because it is depending on experimentation to obtain information, to obtain evidence, to develop theories, because it's natural sciences. I hope it's clear for you. Okay, let me see two types of sciences. Science is divided into two: experimental science, which is it's a use of experiments to obtain information. They must carry out experiments to obtain what? Information. It's only based on experiment. The another one is applied science. They use scientific research. They use scientific research to meet certain human needs, to develop certain human needs, materials, substance, foods. It's called applied science. It's applied on human needs. For example, if you see these pictures when Professor Gabisa Jeta is studying on sorghum researches to meet certain human needs. Because as to increase safety food of human needs. Okay, scientific methods. What is scientific methods? As you remember in the class and also in previous grade 11 studies, scientific method is, the process by which scientists approach their works, done their works. It is a process by which scientists approach their works or done their works. There is a many steps in scientific methods we have. The first one is observations. This observation and asking questions. You must start scientific study by observing and asking questions about a problem. The first one is observation and asking problems. Why? Why is this happening? Why? The second one is do background researches by doing a background researches. By doing a background research. After we are doing background research about the problems, about around that problems, we're going to construct hypotheses. We are constructing hypotheses. Hypothesis means in science it is called scientific predictions or scientific guess. You remember, I hope. And also it's clear. I am presenting in clear ways. Construct the hypothesis, which means hypothesis means a scientific guess or a scientific prediction. Design and carried out an experiment to test hypothesis. Then we design methods and carried out an experiment to test the hypothesis is accepted or rejected. After we are designing and carrying out an experiment to test hypothesis, we are going to analyze the results, the information we are getting from experiments. We are getting information from the experiments. Then we analyze that information called as results. Then we make and draw conclusions from results. We make and draw conclusions from results. After we are making and drawing conclusions, is it our hypothesis accepted or rejected? Accept and reject hypothesis. The seventh one is report results. We are reporting results for those whom it may concern us. Maybe for any government organizations, maybe for non-government organizations, maybe for academic performance in universities, in scholars. So you are presenting wherever you want to present. You present what you are conducting by using scientific methods. So this is all about what is scientific methods. The first one is observation and asking questions. The second one is do background researches. The third one is construct hypotheses. The fourth one is design and carry out an experiment to test hypothesis. The fifth one is analyze result and draw conclusions. The sixth one is accept or reject hypothesis. The seventh one is report results. Okay. In scientific experiments, we must have these two things. One is experimental groups, which is we are conducting experiments. The first one is control groups. A group that is the standard group in an experiment in which we are comparing our experimental group are compared with. This control group is standard, that is, we are controlling this experimental group with. The second one is experimental group is the group in an experiment which is being experimented in order to compare with control groups to get what's changed to solving a problems, to solving a problem. And also we have cause and effects in scientific methods. Cause and effects in scientific methods. The first one is independent variables, and the second one is dependent variables. These independent variables are variables that the experimenter change to see if this is affect performance of dependent variables. When we are going to conduct researches, for example, if I have two farms, farm 1 and farm 2, then I'm adding fertilizers to farm 2. So the farm 2 is called independent variables because it is changed by experimenters. I'm adding fertilizers to farm 2. So the experimenters changed by the variable that experimenters changes. So now I am changing the farm 2 is by adding fertilizers. If it is performances affect the dependent variables. As our expectations, the farm which have fertilizers have more productive than dependent variables. So the farm which have variable is the farm which have fertilizer is called independent variables, and the farm which don't have fertilizer is called dependent variables. These dependent variables are factors in an experiment that scientists measure to see if it changes when independent variables change. We see it changes when independent variables changes. This is about cause and effects, comparing these two things. If the independent variable is good productive, the dependent variable is less than independent variables. We see the changes between these two things. Between these two things. So the another one is controlled variables. These control variables are factors other than independent variables. We have a factors which is other than independent variables that are keep constant in order to avoid influencing the results. We have different factors we are controlling to keep constant our independent variables. Unless otherwise, if we are not controlling our controlled variables, our result is not become good or acceptable, so not valid. We must control variables. Control variables. So another one is confounding variables or extraneous variables. These extraneous or confounding variables is not controlled as controlled variables. It is a factor that cannot be controlled and which may influence result of experiments. We are not controlling these confounding variables. We are not controlling these confounding variables, but it may influence the result of our experiments. It may influence the result of our experiments. This is called variables called confounding variables. The factors that cannot be controlled. It may affect or influence the results of our experiments. The results of our experiments. The another topic is accuracy, reliability, and validity in scientific experiments. We have accuracy, reliability, and validity in scientific experiments. In science, our researches must be accurate, it must be reliable, and they must be valid in scientific experiments. Without these three things, our scientific method or experiment is not acceptable, not standard. Let me see one by one. The first one is accuracy. This accuracy is how precisely something has been measured or counted. When we are carried out scientific experiments, we are measuring and counting something to get information. So how it is precisely or correctly measured or counted. You will see this very well. This level of accuracy must reflect the magnitude of what we measure, or the accuracy of what we measure correctly. For example, this material is the material we are measuring or counting something. For example, syringe. to measure liquids. Reading a burette, these have a measurement numbers. There is a burette, which is also numbers. Reading measuring cylinders, reading a pipette. Pipette is measuring something or counting. So unless we are counting something, we must count accurately or precisely. This is one of the criteria for scientific experiments. The second one is reliability. Reliability means the truthiness of our researches or our experiments. This reliability is the measures of how dependable and consistent the result of an experiment are. The consistent and the result of an experiment are. To increase the reliability of the experiments, our research or our studies must have these three things. The first one is standardize the procedure to always say some things. We must using standard procedures to make our experiment is reliable. To say is it research is reliable? Yeah, because I'm using standard procedures to do always the same things. Repeat it many times. 2. Repeat it many times. 2. Number 3, not try to use. Number 2, repeat it many times. When we are calculating or counting or doing experiments, we must calculate an average result. After repeating many times, 3 times, more than 3 times, and we are taking an average result. The third one is not try to use personal judgment. Instead of using a personal judgment, please count or measure things or that experiments. Don't try to use a personal judgment. This is 3 importance to increase reliability of an experiment. To increase the reliability of the experiment. For examples, we can use special apparatus. That apparatus is called calorimeter, which measures how much light is passed through liquid. How much light is passed through liquids. It is nearly always better to measure than to judge. So, as you say, don't try to say need or use personal judgment. So, it is nearly better to measure than judge. So, if you need to now what light is passed through liquid, we must measure by calorimeter instead of judging. The third one is validity. This validity is whether or not our experiment measure it is measuring. What we are measuring is measuring by another person. For our experiment to be valid, we must be certain our results are only due to what? Change in dependent variables and nothing else. We are focusing on change in dependent variables. So, had we not controlled all over the variables, if we are not controlling all over the variables or controlled variables, our experiment would not have been valid. If we are not controlling controlled variables, our experiment is not valid. We must control controlled variables. Controlled variables. You see, to be our experiment is valid, we must control our experiments. Now, let me come to how do we write reports on scientific experiments. Now we are conducting experiments, but now we're going to report these experiments. The first one is there must be contained. The first one is titles. Title means the heading or ርዕስ matado ርዕስ in afaan oromoo ርዕስ in አማርኛ. Title means which states clearly what is being investigated. It is talk about state. We're going to study about these titles. It is clearly being investigated, which states clearly what is being investigated. The second one is hypothesis, which is stated clearly in terms of how dependent variables expect to influence dependent variables. Often, it is extended to be prediction for particular experiments, guess or A hypothesis is a guess or prediction for particular experiment. So we have saying for a hypothesis, prediction or scientific affection to tell about how independent variable is expected to influence dependent variables. The third one is experimental procedures. We are clear description of our experimental procedures. When we are going to study the researches, how we are going to these experiments, with whom we are going to conduct these researches, with what we are going to research. We must talk about our materials, and this it must be in such detail that anyone of some level of understanding could easily replicate procedure, and it must be included, such as you must know the apparatus you want to use. You have using different apparatus or instruments, starting from pen, pencils, different materials, cars, and also you talk about detail of any chemicals used. What chemical are you going to use after selecting your chemicals? What volumes, what concentrations, and what mass were used? And also detail of any organisms used. Which organism you're going to use, or yeasts, or mice, or what strain of bacteria, and how many you are going to use. And also you now detail of any control experiments. Researchers have a control experiment, it must be become a standard. You must now your control experiments. After we are knowing these three things and you are conducting your experiments, now you have full accounts of the results information obtained by these researchers. You have full account of results obtained, or it is often helped to summarize, although these were appropriate in graphs, in charts, in tables. We have results in graphs, in charts, and in tables. Because when you are finishing or getting your results, you are presenting in the forms of graphs, in the forms of charts, and in forms of tables. Because as it is clear to understand for your audience or for whom it's your present your results. Another one is, or the fifth one is, after you are finishing your results, the conclusions that have been drawn from the results. We are concluding from your results. You are writing conclusions. After you writing your conclusions, an evaluation of the process is held on. It means honest assessment of limitation of procedures that has been used. What is the limitation of my experiments? Point out any avoidable limitation in accuracies, that's errors. After finishing evaluation of your procedures, lastly, you write acknowledgement of use of any other persons. If you are using any other person workers, procedures, you write a person. Those who help you in finance, in idea, in different substance, you acknowledge at the end of your researches. So you put acknowledgement as your reports when you are reporting for others. Now, let me come to the second topics, the tools of a biologist. The tools of biologists. So as you know, biological information is gathered through observational experiments, those conducted in the laboratory and in the fields. Biological information is gathered through observation experiments conducted in laboratories and in the fields. So practical interaction with biological objects in the process are important in making theoretical knowledge more concrete and meaningful. So the biological interaction or experiment, which is biological objectives in the process, are important in making what? Theoretical knowledge. Theoretical knowledge is developed from experiments. If you are conducting these experiments and making these theoretical knowledges made more concrete and meaningful. So many we have many techniques, tools, or instruments are used in biology science. For convenience, they can be divided into tools in laboratories and in open fields. We are using biological instruments into two ways or places. One is used in on laboratory only, and the other one is used in open fields outside laboratory. Hence, in this unit, you will study the use of many of tools of biologists as described below. Then the microscope is studied in detail. We talk about microscope is studied in detail, but we are talking different tools of laboratories in the and also different tools of open fields in biological sciences as members of fields or tools of biologists as members earlier. Now let me see the different materials. This is laboratory or tools. This is field tools. We are plant press, plastic bags and envelopes, insect nets, secateurs, agar, meters, altimeters, GIS or GPS, traps, kegs, diggers, some field tools of biologists. These dissecting keys, mortar and pestles, pipette, microscopes, hand lenses, petri dish, rulers, centrifuge, water baths, glassware, aquarium, balanced and test tube, calculator and computers, the laboratory tools of biology. There are tools of biology in laboratory science. So in the laboratory, let me see one by one, especially by pictures. As you know, this glass is which is growing or developing plants from tissue. This glass is called petri dish. This also have bacteria called petri dish. This is petri dish. This is petri dish. This is dissecting kits when we are surgerying animals to study their anatomy. It's called dissecting kits. Dissecting kits. This is balance. This is electron microscopes. This is electron microscopes, and also this is compound microscopes. So these three things is petri dish, dissecting kits, balance, electron microscopes, and compound microscopes. Let me see one by one, especially about microscopes. You know, microscope is study about or showing small microorganisms in great size. So if you say about microorganisms, microscopes, you remember about magnifications and resolutions. Magnification and resolutions. Microscope is a precision device used to show objects that are too small to be seen at all with naked human eyes. It's very small, but we see as our naked human. This microscope is most important in biological sciences. It's very most important biological science to study about different microorganisms, detail of cell. This microscope is old technique used in biology, but it's still very important in instrument in biology. The advancement in biology are partly due to invention and improvement, modernization of microscopes. Improvement, modernization of microscopes. It's very important to study about different biological area. After invention of this first simple and crude types of microscopes by Anton van Leeuwenhoek, the advancements were made in the field of microscopes. Different advancements. For example, at the end of 19th centuries, light microscope was made, which is used light as a source of energy to enlarge image of an object and improve its versions. The best light microscope shows 1,500 times greater than their size, or original or normal size. 1,500 times growth. At his high magnification, the image becomes less clear because lens cannot be distinguished between small structures lying close together. So, light microscopes have good magnification but have no good resolutions. The power of the microscope is to scatter image and show more detail is known as resolving power of microscopes. It shows a resolving power of microscopes. Resolving power of microscopes. The microscopes could be categorized into the following groups measures depending on their complexity. We are dividing microscopes depending on their complexity. Let me start from simple one. The simple one is called a simple microscope. This simple microscope is a hand lens. In an example of simple microscope is hand lenses, which is consisting of biconvex lens in supporting frames. This simple microscope is too small. The magnification power of common hand lens is usually 10 times up to 20 times. It is multiplying or growing. The objective is 10 times or 20 times. The first microscopes were made up of a single lens and hence were simple microscopes. It become very simple microscopes. The another one is compound microscopes. These compound microscopes, the microscopes were discovered by especially Anton van Leeuwenhoek. This is a microscope which with magnifying power of two convex lenses, which have eyepiece. There is inside eyepiece there is ocular lens or objective lenses. This compound microscope use light rays come from different circumstances, maybe from light, maybe from electric bulbs. Totally, this compound light microscope is called light microscopes. Compound light microscopes are important to biologists because it allows the biologists to observe many kinds of cells in the single cell organisms. Still they are alive. They are moving. They are motile. Still they are alive. So the compound light microscopes increase magnifying power of light microscopes. They have increasing in size. But we see more and more detail up to certain points. There is a limitation of resolution of light microscopes. So beyond this point is the limit of resolution of these objectives or this compound light microscope is called limit of resolutions. Beyond this certain point is the objects get blurred. It's not clear. Image. The detail are lost. It's not shown in detail. It's dibs. For standard light microscope, the limit of resolution is about 0.2 micrometers, or typically cell is about 10 micrometers across. It shows 10 micrometer across. It shows 10 micrometers as it shows. C, and the third one is electron microscopes. This electron microscope is the most important, the most modern way of electron microscopes. Although these electron microscopes are very useful, and their limit of resolution is restricted by this electron microscope. It is usefulness for studying very small objectives. It have great importance to study very small objectives, such as virus, bacteria, and other individual molecules, even not only life, study about individual molecules. In 1920, this physician in Germany realized that electromagnet could be bend stream of image in some ways that glass lens bends beam of light. But this is a more powerful types of microscopes. This electron microscope is a more powerful types of microscopes which is use a beam of electrons instead of light with a great resolving power, unlike the light microscopes. It uses a greater resolving powers, unlike light microscopes. There is also we're dividing these electron microscopes into two types of electron microscopes are known as electron microscopes that use a beam of electrons. When we talk about compound microscopes, it uses a beam of light to produce magnified image, but electron microscopes use a beam of electrons to produce magnified image. The first one is transmission electron microscopes. This transmission electron microscope allows researchers or students or anyone who is using these electron microscopes to view a structure of cell in great details. Allow researchers to view a structure of cell in great details. The second one is scanning electron microscopes. Scanning electron microscopes. It creates image of the surface of specimens by scanning it with high energy beam of electrons. By using high energy beam of electrons. By scanning it with high energy beam of electrons. It uses high energy beam of electrons. So this scanning electron microscope shows details than transmission electron microscopes because it shows the 3D of the images, 3D of the images. It's very important for showing or in any dimensions to watching a image of an object. It shows in 3D. You see? And also, let me see parts of compound microscopes. We're going to see parts of compound microscopes. These compound microscopes have different parts. It's very important to you now about these all parts of the microscopes. This number one is part is called eyepiece. The number one of the compound or light microscope is eyepiece. The number two is body tubes. The number two of the microscope is body tubes. The number three is arms. The number three is arms. And also the number four is nose piece, which is usually to rotating the power of microscopes. Number four. When we come to number five, it is stage clips. The number five is called stage clips. It is clipping the slides. The number six is about objective lenses. It is objective lenses. Number six is about objective lenses. About objective lenses. Who's telling me about number eight? Talk about number eight or number seven. I left for you because this number 7 is most of the time not found on the modern microscopes, but it have their own functions. I left for you. Number 8 is it is about slides. Number 8 is talk about slides. Number 8. Number 9 is stage. The number 9 of this microscope is stage. Number 10 is coarse adjustment. It's used when we are using the low powers. It's called coarse adjustment. Coarse adjustment. Coarse adjustment. Number 9 is coarse adjustment. Number 11 is controlling light from diaphragms. So light concentrating, diaphragm light concentrating objectives. Number 11. Number 12 is called diaphragms. Number 13 is called fine adjustments. Number 14 is power, which means switch on and switch off. And the number 15 is a base of microscopes. Please try to study the all parts of microscopes and use this for your knowledge. It helps you, not it's harm you. Use these parts of microscopes and study very well. Okay, and also let me study about another laboratory objectives or materials or tools of biology, petri dishes. Petri dish is this material which is round dishes which is specially made from glass or from plastics. These are using in different many ways in laboratories, in fields, in this our biology fields, but it usually to culture some organism. Culture means cultivate or growing as organisms.
On the petri dish, we are often filled with jelly substance called agar that is used to culture bacteria or media or food of bacteria. There are many different agars containing different balance of nutrients. Maybe you have nutrient agars, potato dextrose agar, molar agar, extra. Each types of agar encourage growth of different bacteria. Each types of bacteria have supporting growth of different bacteria or encourage different types of bacteria. By marking green on the surface of petri dish, it is helps to grow bacteria on the side of petri dish. So biologists can estimate by using this petri dish covered by bacteria and how much bacteria is covered in growing on the petri dish, and it uses them to estimate how fast the bacteria is growing and how many times the bacteria is growing in these fields. They're going to study about these things. And also agar is jelly-like substance which is obtained from seaweeds or red algae. This agar is used for culturing microorganisms in petri dish. Used to culture microorganisms in petri dish. To culture organisms needs to grow them under a special condition that are likely to help their growth. Help the bacteria by putting in special condition in this normal temperatures, pH, and everything. We are adjusting condition of bacteria. These petri dish are also used to propagate or culture plant petri dish, not only for bacteria. We are propagating or culturing plants from tissue. This technique of growing plants from just a few seeds on special agar in petri dish is called micropropagation. You see these pictures, we are growing plants on petri dish from tissues by method of micropropagations. It allows thousands of plants to produce from just one parent cell. We produce thousands of plants from one parent cell. All the plant conduct produced are genetically identical. The plants produced by this method is genetically identical, as you know. It is genetically identical. What to do biologists use in the fields? Now let we come to the last topic and the portion of this unit 1, science of biology. What to do biologists use in the fields? What tools biologists use in the fields? Biologists do a lot of works outside the laboratories. They study different areas to find out how the animals, plants, and the microorganisms interact with each other and with their environment. They study how animals, plants, and animal microorganisms interact with each other and with the environment. They find out how the area are changed over time, how it is influenced by human activity. They study out human is affecting the environment by increasing deforestation, globalization, and also increasing global warming. This is studied by biologists. So biologists studies these three things, which involves taking measurement of abundance of organisms in the field. They taking measurements or counting how many organisms found in the field to study their diversity. Taking measurement abundance of organisms in the fields, and also taking sample of the environments. They taking samples from the environments to study in laboratory, like soils, rocks, waters for analysis in the laboratories. They going into the fields and taking samples like soils, rock, and the water for analyzing in the laboratories. And they also collecting specimens for identification and analyzing in the laboratories. They collecting fresh specimens for identification and analysis in the laboratory. A specimen means a sample of animals or another organisms to take or study in freshness, to understand its freshness. And also biologists use different tools to gain an estimate abundance of organisms in the fields. Biologists use a quadrant. There are many different types, but the simplest is just metal square. The example of quadrant is metal squares. The common example of quadrant is metal square, which have square shapes, It is placed randomly. We put on the lands randomly on the ground, and the organisms found inside it are counted, and the number and the types are recorded. This data can be used to make an estimate of abundance of organisms in the area. We're counting the number of organisms inside these metal squares to estimate the abundance of organisms in the area. The use of quadrant is not confined to site on the land. They can be used in underwaters. We are not using quadrants only in the lands. We are using also under the waters. Let me see these pictures. This is a person. He take his oxygens and he put quadrants on the lands. Now he going to count a number of organisms inside these quadrants. So we are using quadrant under the water, not only the lands. Clear? I hope it's very clear. We have also transect line or tapes to use or put quadrant the tapes. Then we are using or quadrants. Using quadrants. The biologists use quadrant show how the number of particular species change across an area. How this number of organisms is changing across an area. To do this, they must lay down tape measures or long group mark of every meters across the area. This tape measure is called transect lines. We are using transect line in these tape measures. They then place the quadrants by the side of transect lines. They then place the quadrant by the transect line and record the abundance of organisms in the quadrant. Now there is a transect line. We put quadrant here and we counting a number of organisms inside this area. So we are repeating this counting a number every 5 meters or 10 meters depending on length of transect line. So this gives a picture of how abundance of each species is changing across area or influenced by human beings. Now we are collecting parts of plant can be collected and kept in reasonable condition for short period of time in plastic bags. We are also collecting plants in the plastic bags for studying different plants. Parts of plant can also be preserved using plant press. We are collecting plant in plastic bags and preserving by plant presses. We are collecting in plastic bag and preserved using plant presses. This preserves forms in the shape of plants for some period of time, and specimens analyzed later. It forms the plant shapes and analyzed later. Whole plant can be dug up and replanted for studying in laboratories, collecting and replanted and studying in laboratories. Due to these reasons, we are collecting plants or preserving by these materials. Friction bands is like cartons, bottom and the tape boards is like cartons. We are using newspapers to collecting and preserving plants. We are using cardboard, which means a carton. These preserved plants, you see, it's not changed, but it's collected before maybe 10 years or 2 years. It's using for homes. And we are using these materials. This is cardboard, cartons, newspapers for preserving a plant. Is it very beautiful? Yeah. So when we are using and also collecting these materials, plants, we are using for homes, for office, and for studying in the laboratories. Study in the laboratories. We are using these materials. You see, preserving plant presses, plastic bags, and different cardboards, newspapers for plant presses. We are using for plant presses. And also, however, animals pose different problems. Because they move, they must be caught. biologists do this in different ways. For example, biologists is to collecting insects. Insects, they counting using insect nests. Especially, you see the students using insect nests and they collecting insects. They're using insect nets. So this is another method of collecting moving animals like insects. There is also another method to collecting different animals. Pitfall traps come in a range of shape and size and landed in the soils. It's called we are using pitfall traps. This pitfall trap is the simple one. It's just made from plastic carton that is sunk inside the soils. We are sunk plastic carton in the soils. And also we are make wooden, keep the darken and the dry of the pitfall traps. It keep any animals that fell into the out of sight of predators, outside of predators. We have also different materials that is or used in our apparatus used in the fields. Let me see theodolite. This is used to measure the height of trees or slope in an area. It measures height of trees or slopes. Now, theodolite is used to measure the slope of the plants, trees, or an area. And also we are using a data logger. This data logger is recording information like calculators. What we are collecting information is recorded by these data loggers. This data logger is recording informations. pH kits. It is used to measure about information of acidity of soils or acidity of waters or acidity of what we are collecting. This is called pH kits. The another one is a flow meter. This flow meter is used to measure the rate of flow of waters. This is a flow meter. It is used to measure the rate of flow of waters. This is called a flow meters. It measures how much water is flowing. The speed of waters is measured by these flow meters. This we are using in the fields. It measures how much water is flowing or speed of waters. So generally, we are using different tools. Not only this, we are also using field microscopes. This field microscope is used to investigate the structure of a specimen in the field while still fresh. We are studying about specimens or sample of animals or different organisms. Study whilst it is still fresh. Not only my field microscopes, biologists is using GPS. It's a global positioning system. This GPS is a global positioning system specially used by geographers, agriculturers, biologists to understand the study about latitude and altitude of the locations. Study about positioning system, about location of the study area is studied by this global position system. So this is the end of unit 1 of remedial biology contents. Thank you for attending this double education channels. Please don't forget to subscribe, like, and share double educations, because this double education is your channels and presented by your teachers. And also, as much as possible, please try to share your friends, classmates, friends in other universities, because it maybe help us a little bit. Never underestimate this video. Please use and take some things from this video. ስለዚህ ደባል ኤዱኬሽን ስለተከታተላችሁ በጣም እናመሰግናለን። ግን እንዳትረሱ ሰብስክራይብ፣ ሼር እና ላይክ በማድረግ ለጓደኞቻችሁ እንድትጋሩ በአክብሮት እጠይቃለሁ። ደባል ኤዱኬሽን የእናንተ ነው፣ በእናንተ መማር የተቋቋመና እናንተን ለመርዳት የተቋቋመ ነው። ስለዚህ ብዙ ተማሪ በተጠቀመ ልክ I'm really satisfied. So please share for your friends. Kanaafuu chaanaalii Dabbal Education hordofuu keessaniif baay'ee isin jaalanna, baay'ee galatooma. Chaanaalii kana like, subscribe, share godhuudhaan yaada yoo qabaattan immoo Komantii jalatti nu barreessuu hin dagatinaa. Maaliif yaadni keessan nu jajjabeessa. Akka fooyyeen nu godha. Kanaaf hiriyaa keessan kan yuunivarsiitii keenya jiru, yuunivarsiitii Jijjigaa jiru, yuunivarsiitii biraarra jiruuf akka shirgootan abdiin guddaan qaba. Haramaa yuunivarsiitii akka shirgootan, akka daawwattan, akka fayyadamtan abdii guddaan qaba. Yaada yoo qabaattan immoo tooppikii isinii dhiyeessinu, wanti isin ingilizii yoo jiraate, barsiisaa waan kana nu dhiyeessi jiraate yaada irratti nu barreessaa. Thank you for attending Double Education Centers. Don't forget to subscribe, like and share and post comments if you have comments, suggestion. I love you. See you in next class by Unit 2, Biochemical Molecules. So in next class, we are going to talk about biological molecules, about inorganic molecules, organic molecules, and also we are working reviewcation for Unit 1 and a reviewcation for Unit 2. So if you have any other comments, ask me. I'm going to do reviewcations from Unit 1. Then we are working together in a class and also in video. Please read Unit 2 and also write comments for me. Please change your chance to change. This is my last advice. See you in next class. Bye. Love you.